what is a SNP? If your mic isn't working, let me know in the chat. Britannia, quickly, what's a SNP?
A single base variation that occurs in greater than 1% of the population.
Perfect, right? Very good. Matthew, is it more common in coding or non-coding DNA?
Non-coding DNA.
Very good. Sarah, why is it more common in non-coding DNA, SNPs? You got the harder question, but give it a go.
Is it because the non-coding DNA doesn't convert into a polypeptide? That's why it's not common. I mean, it's more common.
Second part you want to add there is if it's not converted to a polypeptide, it doesn't actually affect our physical characteristics or our phenotype. And so natural selection can't act on it and choose a favorable and unfavorable phenotype until the unfavorable variation disappears, right? And everyone only has one variant, meaning there's no SNP or poly or many forms of the base, right? Very good. So very good answer. I really like that all you guys are revising. When you come to class, the bare expectation is you revise, and I'm so happy when I hear all of you answering the questions well. Alyssa, moving on to you. Why do we care about SNPs in terms of biology? Why and how can we use them as genetic markers, Alyssa?
We can use them as genetic markers because they are inherited with the alleles, which means that certain SNPs will be inherited with the diseased allele.
Perfect, right? That's exactly what I like to hear, right? So if you have a SNP close by to an allele, as you know, one chromosome is inherited from one parent, another from another parent. And so this DNA would be wrapped around the histone proteins of an entire chromosome. And so when this diseased allele is inherited, so is the SNP. So instead of having to DNA sequence, aka read all 80,000 plus base pairs in this allele, which is expensive, time-consuming, and delays results, we can look at the SNPs. In fact, we look at multiple SNPs that occur around a diseased allele to tell us or hint at us whether this allele is diseased or healthy. Now, last question. Kriti, hopefully your mic is working. What do we call a collection of SNPs, Kriti?
Haplotype?
Perfect, right? And we want to look at haplotypes because we can't just look at one SNP, because sometimes someone might inherit a variant of a SNP that's associated with a healthy gene, but in fact have the diseased allele, right? And as you know, that can happen because of, in meiosis, crossing over, where crossing over occurs right between the SNP and the allele that SNP was meant to be inherited with. They get unlinked and they're no longer inherited together because this entire fragment jumps to a whole adjacent homologous chromosome, right? And so this is just a small point that we need to look at multiple SNPs around a diseased gene to tell us if it is in fact diseased with high sensitivity and accuracy. And we call that a haplotype. And crossing over is a rare instance which can cause a diseased allele and a SNP to be unlinked. Okay, very good. Good job. So we've talked about DNA profiling and sequencing. What is the difference between profiling and sequencing? Britannia, on to you. What is profiling in one line, Britannia?
I think it's based on weight or mass.
Yeah, good. So that's a bit about the method, right? It separates DNA based on size. That's what you can say. Profiling uses technology to separate DNA fragments based on size. Okay, so size, mass, etc. But what's the goal of this? I used a word starting with G that I told you that profiling helps to identify. What is that word? Open question to the class.
Genotype.
Very good, right? When you look at this fragment here, right? When you look at this sample here, you see DNA from a crime scene, individual 1's DNA, individuals 2 DNA. I think even a kindergarten student would tell me this lane looks like this lane, which means similar DNA. So straight away from this analysis, I can tell you that it's most likely individual 2, or in fact, with very high accuracy, individual 2 is at the crime scene, not individual 1. But Matthew, if I ask you, Matthew, what's this DNA sequence for individual 2? Can you tell me the exact order of letters from this image here?
No.
No, so you can't tell me the DNA sequence. That is what DNA sequencing does. DNA profiling simply amplifies DNA and then separates fragments based on size and compares it with certain reference wells, such as this one, to identify variation or genotype. Remember, what is genotype? It's just the alleles that an individual has. And all you can tell me now is that the alleles that this person has is similar or identical to this reference well, and individual 1's alleles are different. So you're simply identifying genotypes. Okay, very good. What about sequencing? I'm sure you all know sequencing, exact order of bases. So let's talk about the 2 steps in profiling. Matthew, what are the 2 steps in DNA profiling?
I don't remember.
Does anyone remember what the 2 steps in DNA profiling are? So this is what, remember last lesson, guys, I wanted you to read through all of the modified notes because we're going to go pretty fast this lesson. Anyone happen to read through it? And hopefully all of you did that over the holidays too. What are the 2 steps?
Amplifying or replicating DNA using polymerase chain reaction, and
Perfect.
the second one is, I think, the gel electro thingy.
Good. Gel electrophoresis, right? Phoresis means separate. Phoresis. Electro means using electricity to separate. Where is it happening? In a gel vat. Okay, good. So you guys need to be able to categorize information. This is the thing with biology, and I see this every single year. You get thrown with so much content that it's up to you to identify what you really need to know and what you can learn once and kind of throw away. Okay, and if you can't tell me the 2 categories in profiling, then you don't understand profiling. Because there's a lot of information to it. I'm sure you'll remember last lesson I spoke about the temperatures, the ingredients within the PCR tube, right? But this is the highest yield information right here. So why is it called polymerase chain reaction? I'll do a question to you. Why is the first step called polymerase chain reaction in DNA profiling? Okay, we'll move on. I'm going to ask you again. Rithanya, on to you. Why is the first step? Yeah.
Isn't it the process of where the DNA is replicated?
Good. Using what enzyme?
Polymerase.
Good. There's a special kind of polymerase called Taq polymerase that we use in profiling, and this helps create multiple strands. And this process keeps repeating. And since it's driven by polymerase, and since it's a circular chain reaction, we call it polymerase chain reaction. Okay. Now, tell me a bit about gel electrophoresis. This is probably the step where students struggle a little bit more, because PCR is literally DNA replication, but sometimes instead of enzymes like helicase, we use high temperatures. What about gel electrophoresis? Let's go to Matthew. Tell me a bit about gel electrophoresis, Matthew. What happens?
Can we get a section of DNA? We put it in gel and we put an electric current through it, negative and positive. It's attracted to the positive end, but the larger the DNA strand, the shorter distance it travels. So therefore, we just order the DNA sequence and get short tandem repeats.
Good, very good, very good. Just the last part about short tandem repeats, we'll talk about that later, but you don't have to have the word short tandem repeat in the answer. So gel electrophoresis does not always have to use short tandem repeats. And for those of you who don't know what that is, you should have read it in the notes at least, because that's literally a couple of pages ahead. But if you don't worry about it for now. Okay, but very good explanation, right? So From step 1, the DNA that you amplified into millions of fragments is then pipetted, right, using that tube into this well here that I'm shading. And that well is actually, it's kind of like a finger impression in the jelly. Okay, so it looks like the image here. Yeah, so these are the wells. This is a bird's eye view. This is a much better view. You can see the wells actually extend deep into the jelly. They're like little slots. And so the DNA is pipetted into there and it sits at the bottom. And then when you run the current, the DNA starts running across the gel. Okay? And it's DNA fragments. And we'll talk about why there's so many different bands in each well that you can see. Does anyone know, before I explain, why are there bands like this? Why are there multiple bands for each well? So if I look here, let me just show you this image here, right? So the well would have been here, here, and here, and the DNA would have been pipetted here. Why are there multiple fragments? You see one fragment here, one fragment here, one fragment here, one fragment here. Each fragment, everyone, is an amplified segment of DNA, a fragment of DNA. Okay? So why do we see multiple bands? Does anyone know? Any ideas, Melissa?
I guess it's because it's like some of the bigger fragments are heavier, maybe.
Yeah, but the way we kind of see it is, let me just show you. Here inside this well was just a cluster of DNA. Kind of looks like spaghetti, right? So a lot of DNA has been pipetted there. The reason that you see individual fragments is because in that tube in PCR, what we also add, that I didn't mention last lesson, I wanted to see if anyone knew this because they have asked one question in the trial exams about it. They add restriction enzymes. Restriction enzymes. So these are almost like little molecular scissors that cut DNA at a specific point. So we can use or add restriction enzymes that only cut DNA between two cytosines. So if they see two cytosines next to each other, they'll cut. Or they'll only cut DNA if they see an adenine and a guanine. We artificially create these restriction enzymes and add them into the pipette tubes. Okay, so let's say we have two people. Let's say we use a restriction enzyme that cuts between two cytosines. Let's say person 1 has an A, T, C, C, T, C, C. That's their DNA sequence. I'm not writing the other strand, the complementary sequence, just for the interest of time. And person 2 has C, 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 D, A, C, C. Let's just say. How many fragments would person 1's DNA be turned into? And how many fragments would person 2's DNA be turned into? So I'll start with Critty. Critty, how many fragments would person 1's DNA be turned into?
Wait, so do I only count the C's? Like,
So you need to imagine these are scissors that only insert themselves between two C's and cut the DNA. So they'll cut the DNA here and they'll cut the DNA here. Does that make sense? So how many fragments of DNA will I end up with, Critty?
3.
Good, right? Britannia, onto you. These restriction enzymes only cut between two cytosines. How many fragments will I end up with for person 2? Four. 1, 2, 3, 4. Right? So then, when we run person 1's DNA through PCR, this DNA will get amplified, this DNA strand will get amplified, and this DNA strand will get amplified. And so, in this well here, when we pipette the entire test tube, we'll transfer it and we put it into the well, there'll be 3 differently sized fragments that run down that well. So we should end up seeing 1, 2, and 3 bands of DNA. Does that make sense? Whereas with person 2 has 1 fragment, 2 fragment, 3 fragments, 4 fragments. Each of these 4 fragments, when put inside the PCR test tube, will all amplify, aka replicate multiple hundreds of thousands of times. And so when we put person's 2 DNA, let's say we pipette it into this well, you'll end up seeing 4 differently sized fragments. And since, just looking at this person's DNA, the top fragment, let's say the top fragment is 2 bases, this fragment is 2 bases, this fragment is 2 bases, this fragment is 1 base, only 1 base. This bottom fragment here, since it's smaller, will run further down the well. Whereas the other fragments, depending on the adenine, cytosine, guanine that they have, they won't all be 2 bases like in my little example here. One fragment will be 808 bases, the other fragment will be 2096 bases, the other fragment will be 300 bases, and so then each fragment ends up in a different position. So if you forget everything that I just said in the last 5 minutes, that's okay, because the key thing you need to remember is that restriction enzymes cut the overall DNA into fragments, and each fragment is represented as 1 band. And you can almost think of each fragment like an allele. Okay? I wouldn't call it an allele in your exams, but you can think of each fragment like an allele because it helps you answer a lot of paternity testing and crime scene analysis questions. So if we go to this example here, with the information that I've explained to you, hit the raise hand icon when you find out who the father of this child is. Give you guys about 10 more seconds. Kriti, can you start us off? What are you thinking when you approach this paternity testing question? I don't care about your answer. How are you approaching the question? What's your logic?
I'm not entirely sure, but I was kind of like checking if the bands matched up with the child and mother.
Good. Now, remember what I told you. Treat each band like an allele. Remember, each band is 1 fragment of DNA. Treat it like an allele. What do we say about the child's alleles? A child inherits 2 alleles, one from one parent, and the other allele is from the other parent. Does that make sense to everyone? So the first thing you do is you match up the alleles between the mother and the child because they must have come from the mother. Simple. Where must the other alleles have come from? If not from the mother, Rithanya, where have the other alleles come from?
Father.
Easy, right? So this top band here, does it match up with dad 1 or dad 2, Rithanya?
Dad 2.
Good. Now, what about this band? This band is not accounted for by the mother because it's not present in the mother's DNA strip or electrophoresis well. So which dad does that line up with? This one lines up with 1, and we'll talk about this very shortly. But what about the next fragment here? This fragment of the child's does not line up with 1, so it lines up with 2. Okay, so then between dad 1 and 2, since dad 2 has more matching bands with the child, we'd say dad 2 is more likely to be the parent. Now in your HSC exams, a band like this will not be present. So a band not like this. A band like this will not be present. This was a confusing band because it actually lined up with dad 1. Right? But in the HSC, your options will be very unequivocal or very clear. Okay, all the bands that are not mums will be dads. And the reason in real life this happens is because there is mutation, there is variation in the DNA. And because of that, sometimes unrelated people might have matching up bands just because of chance. And that's what we saw here, where this band lines up with this dad. This could have just been a mutation in the DNA which led to another fragment that now lines up with a complete stranger, which is this person here. So the key point with this is treat every band like an allele. Every allele that didn't come from mum must have come from dad. That's it. So these SNP analysis questions are very easy. So for SNPs, you need to know what the definition of a SNP is. It's good to know as extension knowledge why it's more common in non-coding DNA. You need to be able to draw a diagram by now. And you should be able to tell me the 2 steps in DNA profiling in which we analyze SNPs. And you should know how to read these electrophoresis images. Do we have any questions at all about SNPs before I move on? Good. So in your head, when you know what you need to know and you answer the questions about what do you need to know, biology is not that hard, right? The hard part about is you get thrown with so much content and you need to do questions to find out what's high yield. But I'm telling you guys what's high yield. So good. All right, let's talk about short tandem repeats, this other genetic marker. For that, I think the notes that I wrote for short tandem repeats are up here. Okay. So guys, short tandem repeats or STRs, who's heard of them before? Can you hit the raise hand icon? Anyone heard of short tandem repeats? Okay. Short tandem repeats are another type of genetic marker. They're like SNPs. Okay. Now, this is a difference. Short tandem repeats, you can think of them as short random repeats. Okay. So they're random repeating sequences of DNA. So if I get your entire DNA, there will be sequences of your DNA that just repeats as a AC, 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 AC hundreds of times. And that's just because of chance. All right? Just because of chance, they're repeating sequences of DNA because we have so many letters that make up our genome or our entire DNA sequence. Right? So there could be another sequence that's GC, GC, GC. Or there could be another sequence that's 3 letters repeating, TCA, TCA, TCA. Okay. And the interesting thing is that everyone, if I lined up your DNA with mine, with another person's, all of us have the same repeating sequence in our DNA. So this would match up with this would match up with this. So person 1, person 2, and person 3, this line, would all have repeating A's and C's from this point to this point in their DNA. For this person would be this big, for this person would be this big, for this person it would be this big. And so what this is telling you is that everyone has the same random repeats, but everyone has a different number of repeats. So if this is me, number 1, I could have 56 repeats of A and C. Whereas if I look at Sarah's DNA at that exact same spot, she might have 189 repeats. And if I look at Kritti's DNA, she could have 267 repeats. everyone has a different number of repeats. Now, some people, just because there are 8 billion people on this planet, are going to have the same repeats as someone else. But as long as you understand, everyone has a different unique number of repeats in general. Now, you might all remember we have 2 homologous chromosomes. Now, what I drew here, each line is representing 1 chromosome. Okay, so what that means is if you have 2 homologous chromosomes, you have 2 sets of repeats. So on homologous chromosome 1, I might have 56 repeats, but on the second homologous chromosome of similar size and shape, I'll also have an A and a C, and it'll have a different number of repeats. It might have 167 on my second homologous chromosome, right? Meanwhile, Sarah's second homologous chromosome might have 1,676 repeats of A and C. And Matthew's second homologous chromosome might have, let's say, 1,698. I'm picking random numbers here to show you it's completely random. So key points here, short tandem repeats are repeating letter sequences in our DNA. Every person has a unique number of repeats, and every person has 2 alleles for each short tandem repeat sequence. Each allele is on one of the 2 homologous chromosomes. So this is a diagram we'll again show you. So let's say this is an individual, random person, and on chromosome 7, there are 2 homologous chromosomes in pair 7, we see at this exact locus a CAG repeat. And so this person was going to have 2 alleles for the CAG short tandem repeat. They'll have a random number of repeats on the first allele and a random number of repeats on the second allele, which is on the second homologous chromosome. I picked 39 and 42 just to make it random. Okay, and then down here, there might be another short tandem repeat gene, and there'll be 2 alleles for it. Genes have alleles, and they may have 53 and then 96 on the second homologous chromosome. Is everyone with me so far? So far you should understand what a short tandem repeat is in terms of its location, how many alleles a person has for each short tandem repeat, and the fact that they're unique. Now, the other very important thing to note is that these short tandem repeats only occur in non-coding DNA. Only in DNA that does not make protein. So unlike SNPs, which are more common in non-coding DNA, short tandem repeats are only found in non-coding DNA. Okay, very good. And this means they don't make any genes. Now, the reason we can use them as genetic markers is very similar to SNPs. If we have a disease gene here, let's say there is a gene right here, and it could be healthy or diseased, after the Human Genome Project, scientists found out that a certain number of repeats can be used to predict whether this gene right here is diseased or not. So made from the Human Genome Project, they found that when someone had a diseased gene at this location, 99% of diseased individuals would have greater than 190 repeats. So if you look at this person's short tandem repeat alleles, right, you see 39 and 42. Matthew, do you think the gene that sits right above this short tandem repeat, do you think it would be diseased or not diseased based on this information?
Not diseased.
Good, right? Because from the Human Genome Project, we found for you to have a diseased gene, you're very likely to have more than 190 repeats. This person only has 39 and 42. So it's a very low likelihood that they're diseased. Okay, and likewise, there could be a disease gene right here. And from the Human Genome Project, we found that when someone has a disease gene here, they have less than 100 repeats in this short tandem repeat. Now you see this person's alleles and they have 53 and 96. Sarah, do you think this person has a diseased gene at this location or a healthy gene at this location?
It's diseased.
Good, most likely. See, remember, when we're looking at genetic markers, they're just giving you a very high likelihood of a gene being diseased or not, right? So based on our statistics, we can say the gene there is most likely diseased. Remember, the only way to confirm it is by reading the DNA sequence and going, this doesn't look right, must be diseased, or this looks right, it's a healthy gene. But that's what profiling is good for. It's good for very quick, rapid analysis of DNA, but it's not completely accurate because it can't tell you with 100% certainty sometimes when a gene is diseased or not diseased. Good. Any questions so far about short tandem repeats? Please ask away. Any questions at all? Good, Kriti, question?
Why does like each gene have a different number of repeats that it must have for like to have a disease?
Yeah, very good. So first thing to know is that these numbers that are inherited are completely random. That's why we've given different numbers for each allele. Some people could have 60 repeats on both alleles. Okay. The second thing, Kriti, is it's not causative. So it's not that, when someone has less than 100 repeats, that causes this gene to become diseased. It's just based on the Human Genome Project and looking at everyone's DNA, they saw a correlation. Right? People with less than 100 repeats at this location had a high likelihood, right? People with less than 100 repeats at this short tandem repeat location had a high likelihood of there being a disease gene here, right? But it's not causative. And the reason we see this pattern where, a low number of repeats could give you or indicate disease, or a high number of repeats could indicate disease, is just like Alyssa said, that the short tandem repeats, since they lie close to the gene, they're going to be inherited together. And so people who inherit a low number of repeats of the short tandem repeat would also, by default, inherit this diseased gene. Right? And that's why we see in the population for certain genes, there's a low number of short tandem repeats for that gene. For other genes, there may be a high number of short tandem repeats, which indicate disease gene. Does that make sense, Kriti?
Yeah.
Good. Does anyone have any questions at all? Any other questions? Please ask away, because I had a ton of questions when I first came across this. And I had to Google search it, and it was a headache. So, any other questions at all? Good. All right. All right, I think you guys are ready to do some questions. We're definitely going to finish. We'll finish 1.5 today. But here's the second question. Hit the raise hand icon once you've got an answer. I'm going to get some water up here, I think. I don't see any hands raised, so if you did raise your hand, just hit it again so I can see. Good job, Matthew. Good, Critty. Good. Danny's got an answer. All right, Sarah, I'll get you to take it away. Sarah, what are your thoughts on this question?
I think it's that too, because a lot of the pairs do line up with that too.
Yeah, so first thing you always do is line up the DNA, all the alleles. Remember, these aren't actually alleles, they're fragments of DNA. Remember, allele is a sequence of DNA that makes a protein. These are just cut at random locations based on restriction enzymes. So you should never call it an allele in an exam, but think of them like alleles. So each fragment or allele that was inherited from the mother is inherited from the mother. So we write an arrow and we cross those out. And so the remaining alleles, which I'll highlight, this, and this must be inherited from the other parent. We can see they match up pretty well with that too, right? Let's see, match, 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 match. Easy. So this is how a question would typically be in your HSC exams. This would probably be like question 5 or 6. You can see you can do it in like 10 seconds. Any questions about this? Now, usually they'll be analyzing short tandem repeats when they do profiling. They'll be analyzing some kind of genetic marker, right? And so every person's well could be one short tandem repeat that they're analyzing. And so everyone has a different number of repeats. And the DNA is cut at random locations, and so it creates all these fragments. Just remember from profiling, we usually analyze genetic markers. So short tandem repeats are often analyzed, and hence why Matthew mentioned in electrophoresis, we're identifying genotypes of variations, and he mentioned short tandem repeats because they can be used in this. But sometimes, remember, we don't even have to use short tandem repeats. We can just analyze random sequences of DNA. Good. All right. Let's talk about DNA sequencing. Now, key thing for DNA sequencing and profiling is you need to know the method and you need to know the differences and similarities. Exam questions love asking that. Differences and similarities of sequencing and profiling. So can you hit your raise hand icon? Who's learned profiling at school? No one. Who's learned sequencing at school? DNA sequencing? No? Okay. We'll talk about DNA sequencing. So, I'll get you guys to all go to in your notes. Actually, I'd like you to do this question first. This question was a pain. 2020 HSC SNP question with a pedigree. I'm going to give you all about 2 minutes. Treat this like your trial exams. This is question 18. Have a read and we'll talk about it. Who's got an answer? Who thinks this question is a bit challenging? Hit the raise hand icon. I would hit the raise hand icon if it was the first time for me seeing the question. And I think 99.99% of other students would be. This is exactly what happened in the HSC exam. Do you see how they test you in the HSC? You learnt about SNPs, you learnt about pedigrees, and I'm sure you all knew a lot about it. But this question didn't test how much content you knew, it tested how well you could apply the knowledge and critically think. This is a learned habit as well, how to think hard. And that's why I want you guys to, or every single question I give you, treat it like the HSC exams. Do that in actual exam. Have a very logical and systematic way to break it down. So this question is a lot of information. Let's break it down together. So first of all, DNA from a crime scene was collected ages ago, meaning back then there was no DNA analysis. Okay, so that's what we're understanding. Crime scene, DNA was collected, but we couldn't really do any SNP analysis back then. Point 2 is stating, so I'm going to tick it for I understand that bit of information. Point 2 is saying recently, the crime scene DNA was analyzed at 700,000 SNP locations. Right? That'd be a huge haplotype. Remember, a haplotype is a collection of SNPs. And haplotypes give you a lot of information about things. We're going to talk about them now. So pretty much we've analyzed them at 700,000 SNP locations. A SNP profile was created and uploaded to a genealogy database. Now, SNP profile is just a collection of 700,000 SNPs every person has. So my 700,000 SNPs would be different to Alyssa's, would be different to Sarah's, would be different to Matthew's, right? And that's because we're not biologically related. But then it says, the SNP profile from the crime scene indicated shared SNPs with 2 individuals. Individuals 1 and individuals 2. Okay, the pedigrees for those 2 individuals were constructed. Now, what you're seeing, everyone, is literally how we solve crime mysteries 40 years after the crime has been done. This is exactly what's done. We get the blood, we get 700,000 SNPs, put it on a database, and if on a genetic database we find any 2 people who have related SNPs to that person, we'll create a pedigree and find the link between those 2 people. Good. So now they're asking you, who is the most likely suspect? Right, they put the investigator to be careful, right? But technically, the suspect is the murderer, or he's the victim, whose blood was present at the crime scene. So let's just imagine this is the murderer. So who is the suspect? Can someone break down all this information? What is this question actually asking? If you can filter through all the noise and find essentially, in the simplest way, what do you need to do to answer this question? Anyone know or understand? Okay, so there are 4 different suspects. There's A, B, C, and D, right? There's a crime scene. We've collected SNPs, and we found that person 1 and 2 have DNA in common with the suspect. So you need to ask yourself, out of these 4 people, who has DNA that would be in common with both person 1 and with person 2? That's what this question's asking you. Right, the whole shared SNPs, all of that makes students very confused. Because they're like, oh my God, this is so much information. I barely know what a SNP is. 700,000 SNPs. It's complicated. But when you filter all the noise, essentially what the question's asking is, blood scene, at the blood at the crime scene, DNA was collected, and we know that the suspect has shared DNA or shared SNPs, SNPs are DNA, with person 1 and 2. So ask yourself, who is biologically related to both individuals 1 and individuals 2? That is what the question is literally asking you. So I'm going to give you 30 more seconds to tell me who is the one person out of all these 4 people who is biologically related and thus must have shared SNPs with person 1 and person 2. Hit the raise hand icon once you've got an answer. Nice. Okay, good. Got one answer. Good. But do you see when you can break down the information? The question's actually very easy. Good. I'll give everyone about 20 more seconds. I might just jump into it now because we've done a lot of talking for this one question. Critty, person A. Let's break it down systematically, right? We're going to go A to D. Critty, is person A biologically related to person 1 in this pedigree? Do they share blood with person 1?
No.
No, they're literally married into the family, right? This is a complete stranger who came out of nowhere and just married into the family. So straight away we can cancel A out. Make sense to everyone? How would a complete stranger A have shared SNPs with person 1? Makes no sense. They must be blood related or genetically related to have shared SNPs. On to B. Is individual B related to person 1? Let's go across. Ritanya.
No.
No, right? So individual B had a child, which is person C, who married into the family, right? This individual, if you track up, has common relations with person 1, right? But not individual C. Oh, sorry, not individual B, whose child is individual C. So we can guess that B as well, and that we're already 50% closer to the answer. Option C, is individual C related biologically to person 1? Matthew?
No.
No. So again, you cancel out C and you're left with D, right? But let's logically break it down. Individual D has parents, and this parent came from this parent, and this individual came from this great, how many ever great grandparents? And person 1 also shares the same, how many ever number of greats there are, grandparent. Okay. What about person 2? Right? You can see person 2 came from this parent, which came from this parent. Question tracks all the way up to this individual here who had 2 marriages, 2 sets of children. The other child they had married and eventually became person C, who then had the child person D. So person D is related to both person 1 through this original set of parents and to person 2 through this individual here. And so your answer is D. Not too bad. So you don't have to use any slip. This is literally more of a pedigree question. But you'll find in the HSC, they'll mix and match topics because it's a 3-hour exam, 100 marks. They have to ask you as much content as possible in about 30 or so questions. And so they'll mix content up like this. So do be ready for that. Any questions before we move on? Good. do that question in your own time. All right, what we might do now is I'll talk to you very quickly. Should I give you all your break and then we'll talk about DNA sequencing? And then we'll talk about the 3 use cases of DNA sequencing, okay? It's a bit of a nasty picture, but it's to show something very important. But take your break. We'll recap at about 3:03, 3:04. And then we'll go through DNA sequencing and the 3 use cases of DNA profiling and sequencing. And ideally, it'd be good if we start mod 6 as well. Okay? Any questions before the break? Good. All right. I'll see you all in a couple of minutes. Okay, when everyone's back, you hit the raise hand icon. We'll continue. Cool. Does anyone have any assessment tasks coming up for biology? No. Okay. Usually task 1 is a modeling task. Task 2 is usually some kind of exam, usually in-class exam. So I think this is usually the time where people who know more, where people who've done more questions, it really shows. Good. All right, let's talk about DNA sequencing now. So let me go up and show you which page I'd like you all to be. All right, can you all go to page, there's 43, but it's 156 of your notes. A couple of pages up to here. Okay, now DNA sequencing in terms of use is much less limited, but it is increasing. So like I said, it used to be very expensive, millions of dollars to sequence your genome or your DNA. Now, if you have a spare $500, which most people do, you can sequence your entire genome. I'd probably say wait a little longer and it's going to get even cheaper just because of the efficiency of semiconductors and computer technology. So coming to this piece of information, the method for DNA sequencing is a modified method of profiling. So going to Rathania, what are the 2 steps in DNA profiling?
PCR and gel electrophoresis.
Very good. In DNA sequencing, it's just a modified version of PCR and a modified version of electrophoresis. Now, I'm going to show you in detail how DNA sequencing works. I just want to preface, you do not ever need to explain it to the detail I'm about to teach it to you. I'm going to explain to you once, just so you understand it. Then afterwards, all you need to do is give me 2 sentences on DNA sequencing, just outlining the technology used, and you'll get full marks. Okay, so let's start with the profiling section of DNA sequencing. Just pay attention so you understand it. Remember, from an exam point of view, this is high yield. This is more so you're confident that you know everything in the syllabus and a bit of information outside of the syllabus. There are many versions of DNA sequencing. This is not the only method. In fact, the method that I'm about to teach you, named after a scientist by the name of Fred Sanger, is an old type of sequencing. Now we have what's called next gen or next generation DNA sequencing. And that uses much more complex technology. We're not going to talk about that. Just know there's Sanger sequencing and next generation DNA sequencing. I'm going to take you through Sanger sequencing very quickly. So in Sanger sequencing, we undergo DNA profiling. So we undergo the PCR step. But in the tube, in addition to normal nucleotides like A, T, G, and C, we also have what are called chain terminating nucleotides. Chain terminating. Now, these chain terminated nucleotides are also fluorescent. They do 2 things. I'm going to show you them in different colors. So I'm going to put A in this color. We're going to put T in this color. We'll put G in this color. We'll put C in this color just to keep it. Actually, let me use a fluorescent color for you. Let's pick pink. C. Okay. So I want you to imagine that we are trying to determine the DNA sequence of this strand of DNA that goes by A, T, and C. Okay? So what's going to happen is when this DNA replicates, we're going to replicate and make normal replicates of this DNA. So we're going to make more sequences of A, T, and C. But just because we have these fluorescent chain-terminating nucleotides, by chance, what can happen is when replicating the DNA again, when you have an A, T, and C, instead of a normal, so what binds to an A? T. What binds to a T? A. What binds to a C? G. So now instead of a normal thymine here binding to this adenine position here, we can have a fluorescent thymine binding to the adenine. Now remember, there are two properties of these fluorescent nucleotides. They're fluorescent, meaning they shine bright in a specific color, and they're also chain terminating. So the second they bind to that DNA position, what happens is the rest of the chain stops. It terminates, and you end up with this sequence of DNA that's prematurely terminated because of this fluorescent chain terminating T. Now again, when this original parent sequence of DNA replicates one more time, an A, a T, and a C, we can have a normal thymine binding just because of random chance. And instead of a normal adenine binding in the second position, we can have a purple fluorescent adenine that binds in this position. What happens to the rest of the chain? Britannia? When a fluorescent chain terminating nucleotide binds, what happens to the rest of the DNA sequence?
Would it get like cut off?
Yeah, good. Gets
Or
cut off or
terminated.
terminated. That's what chain terminating nucleotide means. So it terminates. And again, when this parent DNA replicates one last time, just because of, again, random chance, we have an A, T, and a C. We can have a normal thymine binding, a normal adenine binding to the second position. But instead of a normal guanine, we can have a fluorescent chain terminating version of guanine that will again terminate the rest of the chain. So now forget about the normal fragments of DNA. We don't care about them because they don't fluoresce, so they don't shine a color. So they're just going to be invisible for the rest of this technology. We have 3 fluorescent fragments of DNA. This is from the modified polymerase chain reaction. The only difference is we've added these fluorescent chain terminating nucleotides. Now, what was the second step in profiling, Matthew? So the first step was PCR.
Gel electrophoresis.
Good. So in DNA sequencing, we use a modified version of electrophoresis called capillary electrophoresis. It's called capillary electrophoresis because instead of a huge vat of gel, we use a very, very, very thin tube. And those of you who might remember transpiration stream theory from plants in year 11, remember capillary forces are just forces between a thin tube and a molecule, right? And when you have a very, very thin tube, you notice this unique phenomena where water can move against gravity by climbing up the wall of the tube because it bonds to that tube. That's what capillary is referring to. But just remember, second step, capillary electrophoresis. So we use a modified PCR. And the second step is capillary electrophoresis. Electrophoresis. Now, this is kind of like a shoot, kind of like a, you've seen those shows about fiber grades where they, slide down the pole. It's kind of like that. It's a little tube. And the end of the tube is a laser. And there's also computer technology which is going to detect the information. So this is the computer screen from the laser. Okay, so now what we're going to do is we're going to put the solution that contains this fragment here, and this fragment here down the capillary tube. Now logically, Kriti, which fragment, 1, 2, or 3, will reach the capillary tube first? Or which will reach the bottom of the capillary tube, and which one will reach the laser first? The small single base fragment or the heavy large 3 base size fragment?
3 base.
Good thinking, right? You might be thinking heavier things will fall down faster, but this is very similar to gel electrophoresis. They move down by a charge, right? So there'll be a negative charge here, there'll be a positive charge here. So again, think of this like electrophoresis, where tiny fragments run faster and they run further down the tube, right? So with this information, Kriti, which fragment will travel down first or the fastest?
Base 1.
Yeah, right. So fragment 1. So fragment 1 will travel down to the end of the capillary tube first because it's the smallest fragment. And when it hits the laser will hit the fluorescent base and cause it to shine a specific color. What color will it shine? It will shine blue because it's been tagged with a fluorescent blue color. So when the first fragment reaches the bottom, it's going to shine a blue color and it's going to get picked up by the computer. And the computer generates what's called a chromatogram, basically a color detection. And it will detect this big blue jump in the chromatogram. And that will tell the computer that there is a thymine at the first base position because it's been generated, or the computer's been programmed to pick up blue equals to thymine. Very good. Alyssa, which fragment will hit the bottom of the capillary tube next? So first it was fragment 1. What's the next fragment to reach the bottom?
Fragment 2.
Good. And what color will it fluoresce when it hits the laser?
Purple.
Good. So you'll see a purple jump on that chromatogram, and the computer is programmed to pick up purple as adenine. And then finally, Sarah, what's the last or the heaviest fragment that will reach the end of that capillary tube last?
Fragment 3.
Good, right? And what color will it fluoresce?
Orange.
Good. And the computer is detected to pick that up as guanine. And have a look at what we just created. Right? We've created a chromatogram which the computer can use to tell you the first 3 bases in this DNA fragment are T, A, and G. And you can do this. We just did it for 3 base positions, 1, 2, and 3. You can do it for 18 million base positions. You can do it for 1 billion base positions. It's just a matter of time. You look down, this chromatogram is what the computer picks up, and it will convert that based on the color it sees. Since it sees a black, black, and a black, it picks it up as G in this case. Sees 2 blues, computer detects that as C. Sees a red, picks it up as T. You can get the entire sequence of DNA. Just think about this. This isn't cool science or weird science that random people made. This is human thought and critical thinking that led to a technology which we now use to save countless lives. This is DNA sequencing. Now, like I said, you will never, ever be required to explain it to the depth that I did. All you need to do is understand that you know how it works, and if you were to read notes on it, you could figure it out again. But all I need you all to do is give me 2 lines to outline what DNA sequencing's method is. So I'm going to give you all about 30 seconds to say it out loud to yourselves while you're muted. And I'm going to pick 1 or 2 people to give me their 2-line outline DNA sequencing. Imagine it's a 2-mark question. Outline DNA sequencing. 30 seconds, and I'll ask some of you to tell me what your answer is. I remember the first time I actually learned this. No one taught it to me in high school, so I actually learned this first in first year of university. The first time I learned this, I thought it was so hard. So if you feel that way and you feel like this is complex, it is. It's only because it's been festering in my mind for 6 years that I can kind of grasp it. And so I think the most important thing with bio, especially with these complex topics like sequencing, pregnancy, and hormones, the longer and more consistently you think about it, the better you understand it versus rote learning and cramming. Good. I'm going to pick 2 people at random. Let's go with, all of you have been answering very well, so I actually don't want to ask you guys, but Sarah, give it a go. What's your 2-liner for outline DNA sequencing? What would you say in an exam?
So I wrote DNA sequencing refers to identifying the order of the bases. It's basically where the DNA strand undergoes modified PCR to determine the terminating chain. That's what I wrote.
Okay. Good. But do you agree you missed the capillary electrophoresis part?
Yeah.
Right? So that's the other part. So basically ask yourself, it's 2 marks. What 2 things are you going to mention? Right? So Rithanya, your turn.
I said that DNA sequencing undergoes modified PCR, which attaches to fluorescent chain terminating nucleotides, which is then passed down the capillary electrophoresis, which reflects light.
Good. And I think the final thing I would add is to determine the order of bases in DNA, because that's what sequencing is, right? So to get 2 marks for this, I'd want to hear somewhere in your answer, order of bases or DNA sequence to be determined, right? I'd want to hear modified PCR and chain terminated nucleotides, and I want to hear capillary electrophoresis. If you say those 3 things, you've got full marks, right? So an example answer would be, this is why, like, it's cool for you to write it, and this is why I told you guys not to write it, but think of a verbal answer. If you can say it out loud, you can write it, right? And then English can teach you how to write fast, because then all you have to write essays in 40 minutes and all of that. For biology, focus on whether you can say it well without reading off notes, right? So DNA sequencing utilizes chain terminating nucleotides and capillary electrophoresis to create a chromatogram that identifies the order of bases in DNA. That was 1 line, and I mentioned all the key points. Right? You see how I didn't explain all the fragments and the speed they come down and the laser and the fluorescent bases occupying all the positions in DNA? All of that's not needed. This is why when you learn something, you then have to do exam questions so you know what's high yield. But I'm telling you guys, 2 marks is all you'll need. The key high yield part here is, can you tell me all the differences between sequencing and profiling? So now what I'm going to do is I'm going to go across, and I want all of you to write 2 headings, sequencing, profiling. And you're going to write similarities in the middle, and you can write differences at either end. So just very quickly create that table in your notes. Pretty sure I have a table in these notes too, but I don't want us using it. So I'm just going to go across and ask you all. Ready? Once you've created your quick sketch of the table, what's one difference or similarity between profiling and sequencing?
Profiling is basically identifying the order of nucleotides, whereas sequencing determines the genotype.
Oh, careful. You said profiling order of nucleotides. Do you agree with that, or did you mean it the other way?
Oh, sorry, the other way around.
Yeah, good. No, it's all right. Sometimes when I speak fast, I say the exact opposite, but I know what you meant. Good, right? So profiling identifies DNA fragments based on size to determine genotype. Sequencing determines the order of bases. Good. Rithanya, what's one other similarity or difference? Give me anything. Britannia?
Undergoes PCR.
Is that a similarity or difference?
That would be different.
Does sequencing undergo PCR, Britannia?
No. Oh wait, yes.
Yes. Good. Modified PCR. Does profiling undergo PCR?
Yes.
Good. So then a similarity is method, right? Both use some degree of PCR plus electrophoresis. But if it was a 6-mark question, for example, you would say sequencing uses a modified PCR with the chain terminating nucleotides, and it uses capillary electrophoresis with computer technology, etc. Matthew, on to you. What's another similarity or difference?
Profiling uses gel electrophoresis, while sequencing uses capillary electrophoresis.
Good, good. So we've kind of put that there. So sequencing capillary with
Oh.
right profiling uses gel. Good, good add-on. Sarah? What's one other similarity or difference?
Both. Actually, no. DNA profiling can be used as a genetic marker.
So DNA profiling, I know what you mean. You mean SNPs and short tandem repeats can be used as genetic markers, and they're used in DNA profiling.
Yeah.
Right? So you could say, okay, genetic markers are analyzed in profiling, right? E.g. SNPs, STRs, whereas entire sequences of DNA, DNA can be analyzed via sequencing. Okay, Alyssa, that's another one. So we're mentioning very unique similarities or differences, right? Like the whole gel versus capillary. Like I said, in your exams, that's not high yield. We're missing some very, very high yield differences. Think simpler. What are some other key differences in profiling and sequencing? Alyssa?
I just heard that profiling determines the genotype based on size.
Good. Yeah. So that's go. Okay. Let me show you guys what you're missing out on, and then you'll be like, oh yeah, that makes a lot of sense. Money. Which one's more expensive? Uses. What are the different uses? If you don't tell me this, then you're not going to get full marks, no matter how much detail you tell me about chain terminating nucleotides or the specific sequences of DNA analyzed, etc. Right? So you need to tell me profiling is much cheaper, sequencing is much more expensive. Right? You need to tell me the use cases of sequencing, which is pretty. What are the Atul, what are the use cases of sequencing, Atul? When do we use it? Okay, maybe your mic doesn't work still. Pretty? What are the uses of sequencing?
In the, oh wait, or is that profiling the crime scenes?
Yeah, it's profiling, but good. Good, you remembered it, right? Crime scene analysis, paternity testing. Good. And what about sequencing? Scientific research. And now it's being increasingly used in disease analysis too, because it's getting cheaper. Okay. Good. Some other use cases of profiling, everyone, in population conservation. We use it in species conservation efforts too, and we'll talk about that very shortly. Okay, very good. We can also do profiling to understand human evolution. So that's part of scientific research. Okay, but we'll talk about that very shortly. But good. Now you understand everything you need to know to answer sequencing, profiling, and genetic marker questions. Any questions so far? Okay, good. Let's move on. So this is just a fancy diagram of Sanger sequencing from your InFocus textbook, but you'll understand it once you have it read. You can do that in your own time. I decided I'd give you all a 4-mark and a 2-mark answer template for profiling and sequencing, and then similarities and differences. So have a look at it in your own time. But yeah, good. I think time would be another one to mention as well. But profiling is much faster. Good. Let's do an exam question before I teach you the last stop point. We've finished mod 5. So good job. Let me just pick some random questions now because you can essentially do everything. A. Hit the raise hand icon once you've got an answer. I'm just going to stop sharing just because I'll need to do working out for this one. So, and if you see my working out, you'll see the answer. I'm going to stop sharing my screen, but the question is literally, how many genotypes would you see for an organism with 4 alleles? Okay. Just stop sharing for a quick moment. Okay, good. Can someone hit the raise hand icon once they've got an answer? Anyone got an answer yet? Okay, maybe we can all do it together. This is a bit more of a maths question. So if you're good at perms and cons, this is good, but I've forgotten perms and cons, so I'm just going to be doing it freeballing with general maths knowledge. Tanya, got an answer?
I got 10.
Okay. Alyssa, do you have an answer?
I'm not really sure.
And go with Kriti, got an answer?
No, I didn't get an answer yet.
Yeah, I think the key thing for this is when you have 4 alleles, you still only have 2 allele positions, right? And now, like, you can do it the long way, which is essentially writing down all the different allele possibilities, right? So if you have 4 alleles, we can just say, for our purpose here, instead of doing capital letters and lowercase letters, we can just do A, B, C, and D, right? So we can, of course, have AA, BB, CC, and DD. That already gives us 4, right? And then we can think about all the different possibilities when we lock A in the first position, right? We can have AB, AC, and AD, which are 3 more possibilities. Let's keep going. We can also lock B as a first position. But now we can't do BA because AB and BA are the exact same. So you can't think about A. We can have BC and BD, which are 2 more possibilities. So I'm already at 7, 8, 9. And then we can lock C in that position. But we can't use A or B, right? Because AC and CA are the exact same thing. Remember, a heterozygous organism doesn't matter if you're capital R or lowercase r, or lowercase r, capital R, right? You just have those 2 variants. So when you have a C, the only letter you can put in is D, so CD. That's 1. Cross that out. When you have D, you can't use any of the previous letters because DA is the same as AD. Remember that time when we locked A in that first position? We already put a D there. So, Rithani, I would agree with you. You'd have 10 different combinations. I know, silly maths question more than a biology question, but this is a trial paper. And trial papers, they'd be like that, right? So you kind of have to have some degree of maths critical thinking skills as well, just from your pedigrees and Punnett square questions. Okay? Good. Oh, there you go. 28. I'm going to give you all about 20 seconds to give me a verbal answer. It's 2 marks. There are 2 things I'm looking for. Bit of a side question. Have you all got your, maybe it comes out in April or mid-year, when do your UAC course options, when can you start putting in your first, second, third, fourth preferences, etc.? Have they come out yet? Are they coming out soon? Kriti, have you got your, how does the UAC preferences work?
I actually have no idea. I'm not sure.
Rithania, have you been told?
I think we're getting it at the end of this term.
End of this term. Okay, right. The thing that sucks about school, this is, I think, one of the big downfalls of school, is school kind of shows you when you work hard, get good marks, right? There's like clear linear correlation. You work hard, you do better in the exam, you get better marks, and marks, etc. They're like the main metric school is for. But once you leave school, you're not all running the same race, which is the HSC that you're all doing right now. And so school also gives you no insight into what the different career pathways are like. You know, what is commerce like, which 70-80% of your peers or yourself are going to be going into commerce, because that's statistically the most common course that someone picks when they don't know what they want to do in life. And then what typically happens is you graduate after 3 years, you go into the corporate world, and there's about 4 different firms that hire graduates, and students have no idea what the starting salary is like, what the job progression is like, whether you get paid for overtime, what the security is like, what the pathways are. So one big thing I'd recommend is you start looking into it or actively talking to people who are in the career that you're in. Those of you who are in medicine, I think or those who are interested in medicine, it's an amazing, I guess, opportunity to ask and pick my brain about what life is like. You can find out whether you really want to do it or not. The worst thing you can do, it's not really that bad, but it feels horrendous, is once you finish a whole degree, you start work and you figure out, damn, I don't like this. It was an idea that was sold to me that I just didn't look into hard enough. I just ran really hard. Now I'm in the wrong place. And hopefully that happens to none of you, but I wish at your time I continued focusing on grades, but also picked out the brains of people who are where I wanted to be. So law students, if you like law, don't use suits as your guide to whether you like law. Really ask people. And you'll figure out there's a lot of cons in most industries, and you've got to figure out whether you're willing to take it on. Anyway, coming back to the content, only because I see this as the biggest pitfall for students once the UAC, etc. comes out. All right. Anyone got the 2 points for sequencing and profiling? Give you a couple more seconds because I went on an aside. Okay. We'll start with Alyssa. Can you give us 1 point about the results from sequencing and profiling?
I got that they both end up with like a fluorescent, like the DNA strand becomes visible, and in DNA sequencing, computer software technology can use that to determine like the DNA to identify the whole base DNA sequence.
Okay, so at a superficial level, we can say order of bases versus genotype based on size. Genotype based on size, right? So that's one difference, and that's a very basic level one, which we can write down. What's another one? So Alyssa did mention one uses computer technology, the other one doesn't, which is good. Computer technology. Nil computer technology, but that's less of the result and more of the method. So I'm not going to give a mark for that. I'll give 1 mark for this. Kriti, what's one other comparison or distinction point you can make?
Because it says results, can we like talk about the uses? Like one results and the
No, that's not the hard thing. Whereas uses is a whole other category, right? So remember, try to answer the question as best as you can. Don't think too hard between the lines. Bethania?
I kind of talked about the uses as well. So
Okay. All right. Compare the results obtained from DNA sequencing and DNA profiling. So this big little point here, this relates more to this question. I think you're all using it to talk about uses instead for A. So we're all still on 1 mark. Matthew, on to you. Compare the results.
Short tandem repeats on profiling. The comparison of short tandem repeats.
Okay, but remember you have to compare that to sequencing. So what would you say for sequencing?
No idea.
Remember, it's a compare question. You don't want to just throw information that you know about that technology. You want to answer the question, right? If I asked you, what's the difference between the results? You can't just give me a random bit of information about the tech, right? Sarah, on to you.
So basically, I spoke about DNA sequencing and how it uses modified PCR and how DNA profile uses PCR.
Okay, I'll pause. Is that results or method?
That's method, right?
Do you see how it's because you know so much that you're not answering the question? And that's the hard part about these exam questions. Can anyone tell me the other point? You just have to think critically, right? It's asking you result. What result or output do you get from the technology? Sure, you get order of bases versus only genotype as bands, right? Right. The other key point, Matthew, yeah?
Is it the whole DNA sequencing is the whole genome, so the whole DNA, whereas profiling is just a section of DNA?
Close, but even DNA sequencing doesn't have to be the whole DNA. That's entire genome sequencing. It can just be DNA sequencing of the gene. So what I was looking for was whether you could tell me another compare. Remember, it's a compare. So you need to say liken something or find distinctions. Is mentioning both technically give you a visual output in results. In sequencing, you get a chromatogram representing the prevalence of each base in the DNA strand. In profiling, the result is a visual output represented as bands on the electrophoresis gel. So that's one point of comparison. This was one point of distinction. And so I think I've successfully compared the results from sequencing and profiling. They both give you different bits of information, one about the letters, one more about the size, but they both give you visual outputs that can then be further interpreted. Okay, so that's what I was looking for. Not uses, not method, not cost. Okay, good. Make sure you guys answer the question directly. Don't beat around the bush. And that's what kind of answer vomiting is, right? When you get a long response in bio and you just say everything you know about a concept, and you often won't answer the question in that example. All right, let's come back to our content because I want us to finish mod 5. So the last dot point is coming up to here. Investigate the use of data analysis from large-scale collaborative projects to identify trends, patterns, relationships. For example, the use of population genetic data and conservation management. Okay, this is probably the most verbose dot point I've ever heard. I think it's a crappy line, and it doesn't really tell you what you need to know. What you need to know for this last dot point for mod 5, I want you guys to write this down, is you need to be able to give me one example of DNA tech being used in number 1, health and disease, number 2, species conservation, Number 3, human evolution. Those are the 3 headings, and for each heading, you need to give me one example of genetic data being used. And the second kind of question, which is a much more common question type you'll get, is they will give you data about koala species SNP analysis, or the Human Genome Project, or species conservation data relating to haplotypes, and you will have to use your application skills to answer that question. Make sense? 3 headings, give me an example, and then answer any data analysis question related to that topic. Everyone with me? Any questions before I go through the examples? Okay, good. All right, now for species conservation, we're going to go through this a bit fast. I want you all to pay attention. Okay, we have the notes, so it'll be pretty good, right? So for species conservation, the issue is so many species are going extinct. We need to know which species we need to provide more conservation efforts for. Now, Ruthania, would you be more worried about a population that has a high amount of genetic variation, or would you be worried about another subpopulation of a species that has very little to no genetic variation?
I'll be worried for the low genetic variation.
Good. And Matthew, why would Ruthania be worried about a species with low genetic variation? What is the worry?
The evolutionary capacity.
Yeah, good. So under selective pressures, there will not be a favorable trait if they're subject to a new selective pressure, right? There's a high likelihood of that when there's no genetic variation. And so this population is more likely to go entirely extinct, whereas this population is more likely to survive. Now, the question is, how do we quantify or get a number on the amount of genetic variation in a population so we can direct which populations need more help in terms of conservation? Which ones do we need to put in an enclosed facility, treat all their diseases, clean them, make sure there's breeding programs where we hold their genetic information and their sex cells in banks in case they do go extinct? Who needs that, and who can just chill and be in the wild? That's essentially the question we need to ask ourselves in terms of biology and DNA. And so what we can do is haplotype analysis. Remember, haplotypes are a collection of SNPs, and they tell you relatedness. If a population, this is what I need you all to write in red, what I'm about to say next, because if you miss this, you get destroyed in the SNP analysis and haplotype questions. The more haplotypes in a population, the greater its genetic variation. And it makes sense, right? If you have more haplotypes, kind of like that crime scene question, a haplotype is a collection of SNPs. If you have more haplotypes, everyone has different SNP combinations. Everyone's different. No one's related. There's genetic variation, right? Populations with a very low number of haplotypes or low number of SNP collections have no genetic variation, and they need more conservation efforts. Okay, and in terms of example, have a look at this and tell me, right? So this is haplotype diversity, giving it to you as a point, but I want you to look at the numbers, okay? So this is, we're looking at haplotypes for, it should be for question 1, koalas, right? Look at location key 1 to 9, forget about 10 to 20. Look at 1 to 9 and tell me, which koala populations should we be worried about? The New South Wales koalas or the Queensland koalas? Atul, this is your question. It might not work, but I saw your chat, put it in the chat box. Quickly, Atul, New South Wales or Queensland koalas? Who are you more worried about? Forget about the mitochondrial DNA diversity. I'll help you out there. In an exam, they're not going to tell you that. They'll give you tons of information, some not important. What's your answer? At least type it. Okay, I guess your mic doesn't work, or All right, anyway. Going to Sarah. So which koala populations are you more worried about?
I think I would be more worried about the NSW koala population.
Yeah, the Sydney koalas are really risking it. Yeah, you're right, right? So the haplotype number is lower. It's that simple. There has been so many repeats or variants of the same question. People find hard every year because they have not picked out the pattern, which is more haplotypes, more diverse. So this is your example as well. Queensland koalas from haplotype studies, we found that Queensland koalas have more haplotypes. They're more genetically diverse. New South Wales koalas, less diverse. And let me give you a reality. A lot of koalas have diseases, right? And chlamydia, which is a sexually transmitted disease, is pervasive in koala populations. I think in Sydney and New South Wales koalas, it's even worse, which is impacting their survival, right? So this tells us that we need to place more conservation efforts on koalas in South Australia and New South Wales versus Queensland. And so that's how we use population genetic info to help us in saving species. We'll stop. You can do the rest of the questions in your spare time, but that is all you need to know in terms of knowledge. Okay? You can read the information about Tasmanian devils and devil facial tumor disease. It's very interesting because it's one of the rare cases where we have cancer that is infectious. This is, I know it looks really bad. I'm not going to keep it on the screen, but that's cancer or tumor on a devil's face. And when they attack each other, it spreads via cancer cells, which is very different to human cancer. But have a read of that in your own time. Good. In terms of health and disease, your key example is going to be SNP analysis in human disease. And your example is going to be the New South Wales Newborn Screening Program. How many of you were born in New South Wales? Can you hit the raise hand icon? Yeah, good, right? So all of you that were born in New South Wales, your heel was pricked 3 times and 3 drops of blood were taken, put on a card, without your consent, but you were a baby, so it's for the greater good. And it was sent to a SNP analysis facility. This is all New South Wales born babies. And we look out for 33 or 34 different genetic diseases that if we miss would cause early death and disability. And we look for those diseases where if we can act early, we can save life. Examples like cystic fibrosis, where giving you antibiotics would prevent the lungs from being destroyed, or phenylketonuria, where avoiding phenylketones such as Coke prevent brain damage and death. And 3% of babies, think about it, for every 100 babies that are born, every day more than 100 are born, 3 of them have one of these conditions. And SNP analysis helps us pick up who is high risk and who needs further diagnostic genetic testing. Because remember I told you, SNPs don't diagnose genetic diseases. SNP analysis can tell us who is a very high risk and needs further testing. And so this newborn screening program in New South Wales uses SNP analysis to pick out high-risk people for further testing. And here are 3 diseases we check out for. Okay, very good. Now, that's the heel prick we're doing on a baby's heel. Good. Now, in terms of population genetics related to human evolution, we'll touch upon that at the beginning of next lesson, but that is the last tiny bit of information before we finish Mod 5. So literally have a read of it in your own time. In the beginning of next lesson, I'll touch upon this and we'll start Module 6. Okay? Very good. Congratulations. We've essentially finished Module 5. We finished 99.9%. You can do every single question in the Mod 5 trial booklet. Okay? Make your expectation to do every question by latest the next 2 weeks. If you don't finish this trial question booklet the next 2 weeks, you're behind, and it's going to hurt your progress. Okay? Good. Any questions, anyone? Any questions at all? How to study? Any of the content we just learned? courses. Good, good. All right. Well, I'll see you all next week. Make sure you've got some kind of timetable, you're being consistent, and feel free to send questions across during the week if you have time. I'll try getting to it, but I've had 12-hour work days, so I get them very late. But I'll do my best to get to it. All right. See you guys next week.